Donc, je vous présente Ming-Kai Tan, avec qui je collabore depuis déjà une dizaine d'années. And perhaps I should speak in English, so Ming-Kai can understand what I say and correct me if I'm wrong. So Ming-Kai is from Singapore. He did a PhD in Singapore that he defended in 2019. And he works on Orthoptera with many different fields, and he's a very prolific taxonomist. But he's in Paris to work as a postdoc funded by Thyssen Foundation. And he's working with, he will explain you what he's working with, but with evolution of behavior and about personality of crickets. So we have been working together for almost 10 years now, and I'm very happy that we are having some time more or less together despite the pandemics. Okay, Ming-Kai, I let you speak.
All right, thank you. I will share my screen. Okay, good afternoon, everyone. Today I'm going to present on my my work in the museum, and as briefly mentioned by Tony, it's about personality as well as acoustics. And I'm looking at a wide variety of species, so I'm trying to see how these personality and acoustic traits evolve in specifically the crickets from the subfamily Eneopterinae. Okay. So Tony has already introduced me, so I will not talk too much about this part. So for the talk, I'm going to separate it into 5 main parts. So firstly, I'm going to introduce myself, introduce my topic. So I'm going to talk briefly about variation, introduce to you guys the concept of personality and how acoustic communications may be related to personality. Then I will go through different levels of variations. So firstly, I'll introduce about individual variations. So you'll be looking at a case study of using 2 species to study the personality as well as individuality in the calling behaviors. Then I move on to population levels. So here I'm more interested in looking at what are the differences in the populations, and given the situation of the COVID, how does that impact the research study? And lastly, I'm going to talk about at a species level. So we have collected data for numerous species so far, and that allow us to compare how calling behavior, specifically how the circadian rhythms of calling behavior differs between different species of Eneopterinae. And of course, I'm only halfway through my postdoc, so there's still a lot of work ahead. And of course, I will try and summarize how does all these different parts tie in together into a bigger question. Okay, so firstly, introduction. I think I have to start off with variation in biology. So variation is simply differences in traits of in the animals and plants in all kinds of living organisms. And it is the basis for sexual and natural selection process to take place. And we humans can categorize these kind of variations or differences in terms of continuous variables as well as discontinuous. So, for example, body mass and blood type are just 2 different kinds of variation that we can relate to. So there are different levels in which variation occurs. So different species have different varies differently in their characters. Within a species, different population can also exhibit different variation. But I'm looking at least for the personality, I'm actually looking at individual variation. So uh, a more familiar individual variation might be phenotypic plasticity. So that is how variation, how a population or individual varies according to environmental influence. But I'm going to talk to you guys more about personality. So animal personality is a rather recent topic, and there are still many debates about how relevant it is to biology in general. So typically animal personality is defined as a consistent inter-individual differences in behavior, and it is applicable in different paradigms of behavioral research. So it ranges from anti-predation, finding mate and making mate choice, as well as foraging for food, as well and learning behavior. So some commonly studied personality traits include shy and boldness. So this is one spectrum that people look at. Another spectrum, which is more relevant to predators, would be, for example, passive and aggressive behavior. Generally, animal personality, people in animal personality research, people would come out with a value that ranges from 0 to 1, 1 being highly repeatable, and 0 means it's not repeatable. So this is a repeatability estimates, and as you can see from the formula, it comprises of between individual variants as well as within individual variants. So for a character or for a trait to be repeatable, there is one possibility is that there is a high variance between individuals, or it could also means that there is a low variation within the individual. So that these are two scenarios in which that can define a trait to be considered repeatable, and hence can be considered a kind of personality trait. And there are different ways to calculate these repeatability estimates. The formula that I've shown is the really basic one, but with the complexity of the models that are available today, the actual models to calculate these values are a lot more complicated. So the paper here that I've shown here is just a really nice paper that try to explains the mathematical aspects of how people develop these repeatability estimates for biologists. So why does animal personality matters in biology? Traditionally, people do not really take into account of inter-individual variations, so they would usually consider that as statistical noise in the models. But nowadays, people are realizing that actually these inter-individual differences can have ecological and evolutionary importance. So, for example, an individual which are always bolder and more aggressive are probably going to be able to find more foods and maybe find more mates. But at the same time, this kind of behavior is going to make them more susceptible to predation. So in a natural population, in a natural world, there is going to be selection for different kinds of personality traits depending on the environmental pressures. And because of that, many of these traits that are being studied in animal personality can be considered adaptive. Another more practical relevance of animal personality is that when people collect wild-caught animals for the experiments, they might actually be collecting individuals that are bolder, that are more exploratory or more aggressive. That's why they have a higher chance of being collected. So that actually might create a sampling bias where shyer individuals and those that are less exploratory may not be actually be collected. So this is something that that biologists might have to consider when they are doing such kinds of experiments. So I'm also looking at acoustic communication. So I want to see how acoustic communication can be relevant in the field of animal personality. So as I mentioned, one of the very common personality trait that people look at is boldness. And boldness can be defined as a tendency to take risks, especially in when an individual is exposed in a novel environment. So a traditional means of quantifying boldness would be done in open fields. So an individual is simply placed in an arena that is considered novel, and then we track and see how they behave. Another way is to introduce a novel object and see how this individual responds to this novel object. And lastly, usually the individual is also introduced in a environment that they are from an environment that they are more acclimatized to, and then we want to see how long it takes for these individuals to actually move and decided to explore the new environment that they are being introduced into. So for species that produce calls like birds and crickets, calling is considered a risky activity because on one hand, it helps to attract the mates. At the same time, it can also attract predators as well as parasitoids. So for individuals that is more willing to call, that is also an indication that there is more willing to take risks. And would that be considered a bonus traits? So that is something that I hope to answer through my research here. So I found some knowledge gap in this area. So I'm interested to know how relevant acoustic is in personality and individual variation. And so far, many studies that works on personality as well as acoustic tend to be focusing on a few model organisms, and we don't really know how widespread this kind of behavior or how relevant this kind of behavior are across different groups of animals. So that prevents us to understand how personality or behavioral traits can evolve. And so with that, I developed broadly three research questions. So firstly, I want to look at how acoustic parameters can be used as a personality traits. And I want to see how these personality traits, not just the acoustic, but the traditional means of measuring personality traits, how widespread they are, and how these personality can have evolved. So for my study, I'm actually looking specifically at the crickets from the subfamily Eneopterinae. So this is a monophyletic group of crickets with a relatively well-established phylogeny. So we know the relationship between different genera and how they are related. But more interestingly, I think is that they are very diversified in their morphology and their behavior. So unlike many typical crickets, many of these species actually produce calls that are at high frequencies, so that can be ultrasonic or near ultrasonic frequencies. And very recently, people has been finding new and unique communication modes that these species use in when they are communicating. So it's not a one-way male calling and attracting a female kind of system. So males would explore around to look for females. Females will do the same explore, and they actually also respond through vibrations. So there is a lot of communication that is just beyond acoustic. So this also means that they can be a very useful model system to study how they communicate in general, not just in terms of acoustic, but also in terms of their exploratory behaviors. So you can see these are just 4 different kinds of eneopterines that we are looking at. So there are the wingless ones. There are species that produce frequencies that are at a normal 5 kilohertz, around the 5 kilohertz range of a typical cricket. And there are also fully winged species, Brachyptus species that can produce calls at high frequencies. So going to the second part of the second aspect of my talk, which is to look at individuals, individual variations. So as I mentioned, we don't really know whether personality exists in Eneopterines. And likewise, we know very little about inter-individual differences in the calling behaviors. For Eneopterines or for animals which actually produce calls as well as explore around their environment to look for mates, they need to strategize, or that there has to be some kind of strategization to make sure that they can find their mates without being eaten by a predator or being parasitized by parasitoids. So it will be useful to study the acoustic as well as the behavior, exploratory behavior of these animals concurrently. But as far as we know, very few studies actually uses such approach. So we came out with two research questions, pretty basic research questions, and that is to determine whether in these crickets is there a consistent inter-individual differences in both the calling behavior as well as the exploratory behaviors, and if they do they correlate? So does exploratory behaviors correlate with calling behaviors? So in this study we use two species as a representative, and one species is the Lebinthus, and the other from Macrobinthus genus. So one of them is a diurnal species, and the other species is nocturnal. So they produce pretty different calls. So this allow us to look at how they are different as well. So how do I quantify the traits? So I'll first discuss, I'll first introduce how I measure the exploratory behaviors. So each crickets are first placed in a glass jar for around 30 minutes. So they are allowed to acclimatize in this glass jar. So I mentioned previously about how they move from, how long they take to emerge from a place that they are acclimatized to a new environment. So this is what I'm trying to do. So after 30 minutes, I will introduce this cricket in the glass jar inside the testing arena. So this arena is considered a novel environment to the crickets. And once that is introduced, our video record how they move, what are the behaviors in 40 minutes. So that includes how long it takes to emerge from the glass jar into the testing arena and other behaviors. So in order to quantify within individual variation, I have to repeat the experiment at least 2 times. So specifically, I'm actually repeating the experiment 5 times for each individual so that I can have a measure of the within individual variations. And of course, we also have to look at the between individual variations. So multiple individuals has to be done. So like I mentioned, I will look at how long it takes for the crickets to emerge out from the glass jar into the novel environment. So the longer the time it takes to emerge out of the glass jar, we can consider it to be less bold or less exploratory. And once it's inside this novel environment, once it has left the glass jar, we also look at how long it spent exploring the new environment. So the longer the time is spent exploring, the more exploratory this individual is. So we are exploring more traits, more traits that we can use to quantify personality. So for example, instead of just looking at the time, we can also look at the distance traveled. And to do so, we need animal tracking softwares, and this has not been done yet. So secondly, there is a part about the acoustic recordings to extract information about the core parameters. So for each individual, we actually record it in isolation. for 24 hours in a sound attenuated room. So this is to reduce disturbance and reduce influence by other males or other calling males on the calling behavior of the particular individual that we are looking at. We are also controlling for the temperature, because we do know that temperature can also influence the call patterns or call parameters of the crickets. And of course, we try to create as realistic environment as possible while standardizing as many parameters as we can. So what we did was also to create this day and night time to see how this behavior might differ between the day and night. So as I've mentioned earlier, the two species that I looked at, they have very different kinds of calls. So for the Lebinthus, there's actually two parts. So you can see on the left panel, there is the part where there is multiple clicks, very isolated clicks, and followed by a continuous trilling part, the trill. So for these species, we can look at the call duration, we can look at the number of chirps, we can look at how the trill duration as well as the syllable period within the trill duration. On the other hand, for Macrobinthus is more simple. So each call is actually just made out of a trilling part. And so in that case, we could only quantify the call duration, which is analogous to trill duration actually, as well as syllables period. So how are these repeatability for these traits being estimated after we have extracted all this information from the videos as well as from the sound files? So I'm using mixed effect models. So in this case, when we are talking about mixed effect models, we always have a random effect. So the random effect in this case is the individual crickets. And by assigning the individual as a random effect, we can actually calculate, or at least the models can calculate the within and between individual variation. And then based on the fixed effects, which are things like the time of the time of the experiment, what trial number it is, the weight of the individual. These are confounding factors that might influence the behavioral traits. So the models can take into account all these fixed effects as well as the random effect to give us a adjusted R value, which is the repeatability estimates. So from our experiments, we could answer the two questions that we set out to investigate. And we found that yes, calling behaviors, or at least the temporal parameters, are generally repeatable, which means that some individual consistently call longer than other individuals, have a longer call duration than others, for example. And likewise, for the exploratory behaviors, we also observe consistent inter-individual differences. So this means that some individuals are consistently more exploratory in their behavior than others. So some individuals being more flamboyant, or means that they are more willing to call, or they have longer call durations, means that females might be able to detect these males more readily, but at the same time, these males are also exposed to predators or parasitoids or even rival males. So likewise, the males that are more exploratory are probably going to be able to find the females, but they could also face more dangers as they explore. But because we have found that these behaviors are actually repeatable, it also allows us the opportunity to explore whether are these behavior correlating. So the question that we want to answer is that are individuals which are more exploratory, are they also more flamboyant? And we use two approaches to do this correlation. The first approach is the multivariate mixed effect models, which means that we have two response variables inside the models. These kind of models allow us to take into account of the variation that is inherited from the individual. So it can account for the within and between individual variations to come up with a more conservative and more reliable estimate of the correlations. We also do a more basic kind of analysis, which is simply to create a PCA, summarize the core properties, and using one of the axes to correlate with the exploratory behavior. But unfortunately, either models or either approach, both approaches show that there are actually no correlation between the exploratory behavior and the core properties. So this means that some individuals can be more consistently calling longer as well as more exploratory. Others are actually less vocal and doesn't call as well, doesn't explore as well. But then there are also individuals that they are either more vocal but less exploratory, or less vocal but more exploratory. So this means that different individuals might probably be using different strategies to communicate and to find mates. And this seems reasonable because, especially in an unpredictable environment that they might be in a natural world, so having different strategies will ensure that this population, the diversity of the population can be maintained. So going to the population levels, we have been carrying out experiments in the lab colonies, So these are individuals that probably come from a few parents and over many generations, and the genetic variability is probably much lower than what is occurring in the natural environment. So we are interested to look at wild individuals and repeating the experiments for individuals from different populations to see whether we can get any new insights compared to our experiments in the lab. So this is where I went back to Singapore, where I collected the crickets from 3 isolated populations. This time around, I'm focusing on another species of Lebinthus, the only species of Lebinthus that is found in Singapore. And I actually technically follow the same approach and same methodology as the previous experiments. But unfortunately or fortunately, COVID-19 pandemic happens, and just like in France, Singapore went into a lockdown for 7 weeks specifically, and the experiment has to be stopped during this period. But because of this, it allows us to actually compare data of that we have collected before the lockdown and after the lockdown when experiments can be resumed. So instead of just looking at how the calling properties and exploratory behavior differs between the population, this time around with the lockdown, we can also look at how are they different between and after the lockdown period. And many scientists are using these opportunities to look at how human activities impact wildlife using the lockdown as a natural experiment. So the first thing that we wanted to look at is actually population differences. So if you look at the graph, if you ignore the blue and the orange coloration, you just focus on the different population, you will be able to see that the trill duration and the core duration tend to be significantly different between the population. We didn't see any significant differences in their exploratory behavior. So we only see differences in their calling behaviors. And why is it so? It probably means that different populations are subjected to different pressure, and that leads them to resulted in microevolution of these traits. And what we can infer from is that in the high heat and the ubin population, where the call duration and the trill duration are actually lower, we can infer that that is because that these sites are actually very popular sites for hikers, mountain bikers, and exercise goers. So it also means that there is a lot of human disturbance and a kind of a disturbance or kind of a human deterrence response from the cricket is to actually shorten their calls. And of course, that is prior to the COVID-19 crisis. But when we compare the call properties before and after the circuit breaker or the lockdown in Singapore, we found that in the high heat population, actually the trill duration actually further shortened after the lockdown. And that is not really the case for the other population. In fact, the other population, there is a slight increase in the trill duration. So why is that so? So during the lockdown, actually the high heat population, which is situated in this nature park or high heat nature park, actually receive even higher human visitorship because everybody is locked down, nobody can travel, but they are still allowed to visit parks like these nature parks. And it happened that this nature park is situated within a dense housing estate right in the center of Singapore. So that means that a lot of people can actually access these nature parks, and because of the restriction, people are actually visiting them in larger quantity, and it has been reported in the news. As compared to the other two sites, so the other two sites are Labrador. It's actually located in the central business district of Singapore. So during the lockdown, where everybody is working from home, the central business district are actually pretty empty, and the visitorships to this nature reserve is probably lower than compared to before the lockdown. And likewise, Pulau Ubin is an offshore island that is actually you need to actually take a boat to reach there from Singapore. So during the lockdown, chances are that there are actually also fewer people visiting these offshore islands. So this study is actually, at least from what we have found so far, is that it's actually the first demonstration of how the COVID-19 pandemic affects invertebrates, affects the behavior of invertebrates. There has been increasingly more papers being published talking about the impacts of lockdowns on animal behavior, but most of them are actually conducted or focused on birds. So, but it also allow us to understand how human activities have been impacting the animals even before the pandemics. So lastly, I'll go into the species level now. Because we have done a lot of recordings, as I mentioned in the previous parts, in the acoustic room in the museum itself. And we actually recorded each individual over 24 hours. And this actually allow us to look at the circadian rhythms of the crickets for different species. So circadian rhythms is an endogenous, generated, and self-sustaining oscillation that occurs around a scale of 24 hours. So is ubiquitous amongst animals. We are also influenced by the circadian rhythms. That is why we would sleep at a certain time of the day, and it influences our activity across the 24 hours. So these are ways in which species strategize for their behavior. So that includes trying to escape from predation and parasites, as well as competitors. And different species may vary in their circadian rhythms, and people has been demonstrating that or arguing that circadian rhythms can be an important contributor to reproductive isolation and sympatric speciation in some animals. So for Eneopterinae, there's actually quite a lot of information about the calling songs. So we look comparatively to many other cricket subfamilies, we do know about the calling songs of many more species of eneopterines. And from these, typically these are sporadic observations or observation in the natural environment, and people are able to classify certain species as diurnal or nocturnal. But these data are not precise enough, not comprehensive enough, and it's not standardized. So there is a lot of factors that might be influencing the calling behavior or calling activities in the field. So by conducting standardized observations in a very controlled environment that we have done, it allows us to see the complete picture of the circadian rhythms of these crickets. So we quantified calling activities using two approach. The first one would be the number of bouts or the number of imps. Imps means that the number of consider one unit or one unit of the calling songs. And so if you look at the pictures, you can see that for example, the panel D, Macrobinthus, this is the same species. So here it shows two bouts from this example. But there is a problem with this approach. So you can see that in the same scale, some species has a much longer call duration than the others. So it also means that the amount of song that's being produced is different for different species, even if they have the same number of bouts per unit time. So that might not be the best way, or that shouldn't be the only way to quantify a calling activities. So instead of just counting the number of bouts, we also look at the amount of sound produced. So we take into account of the call duration or the duration in which sound is being produced from the crickets. And so far we have data for 11 species. And as already shown from previous observations, some species are indeed strictly diurnal or strictly nocturnal. So you can see those graph that is red represent species that are most strictly diurnal, and the graph that are blue indicates nocturnal species. Then there are some species that calls both in the day and night, and their patterns can vary very differently. So some species call for a short period of time, but during this short period of time, they are continuously calling. But there are also other species that call pretty consistently throughout the day and night, but then there are actually some higher levels of activity compared to others. So we looked at both the amount of sound produced as well as the number of bouts, and generally we actually see similar patterns within each species. But there are some exceptions. So if you look at the graph for Nicitrus, for example, Nicitrus vittatus, you can see that based on the amount of sound produced is actually very high, and it actually shows two peaks that is pretty distinct. But this is not the case when we look at the number of bouts, and it's actually very low compared to other species. And the two peaks are actually not that distinct. And we rationalize that it is probably because for these species, it can actually produce a really long call each time. Each bout can be consisting of a very long call. So even though within an hour, they might only produce say 2 bouts of calls, but each bout is actually very long. So if you quantify the sound produced, it's actually a lot of sound being produced. The Cardiodactylus Palawan is also an exception that is interesting, because when we look at the total sound produced, it's actually a pretty consistent calling across the day and night. There's a little around maybe around 1800 hours. But once we look at the number of bouts, we see that there's actually two very distinct peaks. One slightly after dusk, around the 1900 hours, and another one somewhere in midday, which is not observed when we look at the sound. So the reason that we can think of is that probably maybe because at different time of the day, the cricket actually changes the number of pulses within each bout. So even though they might be calling at a very high number, the number of syllables may actually differ, such that actually if you look at the amount of sound produced, it's pretty consistent over the entire circadian rhythm. So like I've mentioned, the citrus species, if we look at just the number of bouts, they're actually pretty low compared to other species. But once we look at the amount of sound produced, they actually produce really high, really large number of sound. Likewise, there are other species that if you look at the number of bouts, seems like they are producing a lot, they're calling consistently all the time. But actually each call is a very short time, it has a very short time duration. So if you consider, if you look at the sound produced, actually they are actually really low, as you can see in the Cardiodactylus situation. So why does these species have different circadian rhythms? Can probably be attributed to their ecology and their behavior, which we may or may not be able to answer at the moment. We can make some inference. So we found that the Lebinthus species in Singapore, they are actually an important prey item in these mammals called the common palm civet. And when we look at the circadian pattern of these palm civets, it's actually kind of reflective of the circadian rhythms of the Lebinthus species. So when the behavior, when the palm civets are actually active, the calling behavior tends to be lower in the Lebinthus species. So we can make some inference, but we can't really be sure because the real dynamics, the real ecosystems are a lot more complex than just a common palm civet eating the crickets. Some species also have very short call periods, as you have seen in the Cardiodactylus species. And these are probably different approach or different ways in which species can avoid predators or parasitoids. So by calling within a short time period and remaining silent for the other times, probably help them to find their mates more effectively without being eaten. And one other reasons that explains this circadian rhythm would probably also tie to the activity levels of the females. So for example, when the females are active, the males might be calling more willing to call, and the calling activities would consequently be higher. So circadian rhythms has been traditionally overlooked in communication studies, as well as studies that look into selection pressure and selection processes. But with this kind of data, and we hope to collect more data for more species, it offers us opportunities to look at the role of circadian rhythms in the evolution of communication systems in these crickets, for example. So this leads me to the last part of my talk, which is what I plan to do, or what are the plans that we had ahead. So we wanted to look at more traits for both the core parameters as well as behaviors. So we are also modifying our experimental approach to look at more traits that is not possible at the moment. We also want to look at population dynamics and differences in the calling behavior along a gradient. The gradient could be disturbance or urbanization gradient. So we have found that population differs in their calling behavior and at different time point, but it will be, I think there will be more insights if we look at more population and we look at we are able to quantify what actually explains this calling behavior or these population differences in calling behavior. And as I mentioned in the introduction, I'm interested to look at how this personality or this behavior evolved in these crickets. We have around 10 species, data for around 10 species, and that is really not enough to really study the evolution of these traits. So the aim is to actually collect more species if that is possible. So in summary, I have 3 different parts. I've talked about 3 different parts, and we show that there are some repeatability in their calling behavior as well as the exploratory behavior. And we also see that we also have found that population exhibit differences in these parameters and this behavior, and as well as how human influence or human activities have an impact on this behavior. And the species, at a species level, we can see that there are differences in their circadian rhythm, but this is what we have so far. And we would like to better understand how core parameters can predict bonus, or how is that related to bonus in terms of personality. And we hope to collect more data for more species so that we can eventually perform some kind of ancestral state reconstruction of the personality traits as well as the acoustic traits. And particularly, we would be interested to look at the circadian rhythm. As I mentioned, it tends to be overlooked. And so we hope to be able to address this. And yep, with that is the end of my talk. Thank you for your attention.
Thank you very much. If there are questions. Yeah, there is Violaine. Do you have a question?
Yes, can you hear me? Can you hear me or not?
Yes.
Yes?
Yes.
I was wondering, so the different species you compare for the circadian rhythm, can you tell us a bit more about their phylogenetic relatedness, and also do they occur in sympatry or are they from different geographic areas? Can you give us a bit more details?
Yes. So for the 10 species that we have recorded, we have actually sampled pretty diverse across the phylogeny. So it includes the main clades or the main groups of Eneopterinae. And so far, we do have species that occurs in sympatry. For example, the Cardiodactylus Palawan, one of the species, is it can be found in sympatry to Lebinthus, both species being found in Singapore. But are they syntopic? I don't think so. So I don't think they are found within the same place within Singapore. But yes, but we also see sympatry in Lebinthus species. So I think there are 2 species that can be found within a single place within Philippines. So there are species that are sympatry, but we also sample across pretty broadly across the phylogeny.
Okay, no, my question was like related to the selective hypothesis regarding this calling hours. I mean, if they are closely related and they live in sympatry, maybe there are some reproductive interference between the species that would promote divergence in the calling hours of closely related species in for instance. So that's the kind of thing you could test, but of course you would need a larger coverage of different species to know the phylogenetic signal on this trait and so on and so forth. Yes. Thank you.
Thank you.
Other questions? Oops. Vincent? Vincent? Est-ce que... Alors, le micro a l'air allumé, mais on n'entend rien du tout. Ah.
Maybe
There
Vincent,
seems to be a question, but I can...
Maybe you can write it down on the chat. Maybe you can write it on the chat, this question.
Ah, okay. I can see it in the chat yet, so I don't know.
Oh, I was just suggesting he could do that if his microphone doesn't work.
Yes. Yeah. Okay. Seems like Vincent has given up. No. Vincent, have you tried using the chat then?
I have Vincent on the phone. He's trying to to communicate with us. So, what is your question, Vincent?
So actually, it was not really a question. Can you hear me?
Yes. Yes, he can.
Yes. Okay. So sorry about that. I don't know why it's not working. It looks like I don't have the right to ask my question on the chat. It's not working for me. It says that the administrator should allow me to do something. So anyway, so it was not a question. It was more a remark related to violence question on the circadian rhythm and stuff like that. So I would expect that when there are predators around, it would be maybe advantageous for the different for most individuals in sympatry to sing all together, so that to be sort of protected by the global noise. And so I was wondering if there was any situation where you would be able to test that, to contrast situations where there are predators, and then you would expect a somewhat synchronous synchronization of the calls, while in situation where there are no predators around, or maybe less numerous predators, then you would expect that the sexual selection and the competition among species, among individuals from different species, would prevail, and with a separation, a temporal separation of the calls. I mean, that was just an idea like that, but I don't know if you have any data that would allow you to investigate that kind of things.
Yes, I think the experiment that we did is in the colony, is in a in situ lab-based kind of experiment. So actually, we do not really know how does this how are these relevant in the natural environment where there are actually more than one males calling, and also like you have mentioned, the absence and presence of the predator. So one way that we hope to look at as well is to actually carry out the recording in a field, so so that we can actually not only record a single individual, but actually looking at the entire population of the crickets in a natural setting. And then hopefully from the recordings, we can also identify the presence of the predators. That would actually help us to answer this question better, as well as if there are more than one species co-occurring at the same time, how do they synchronize and in their calls that can help more than one species to avoid the predators. So yes, that is something that would be nice that we can, if we can actually explore further.
Okay, thank you, Ming-Kai. I have to go, but it was a really great talk. Thanks again.
Thank you.
So any other question? Well, then thank you all very much. Thank you very much, Ming-Kai. And it was really great. Have a nice day, everyone.
Nice day. Thank you.
Thank you very much.